Thank you, Professor Mario Ros Patrizio, for a very elaborate introduction. I feel my introduction is only a student in foraminiferal research. I'm still trying to learn many things, which I could not do so far. It's not that we are the pioneer in this field using foraminifera in pollution monitoring. Many people are working all over the world. But today I'm going to share my experience working along the Western continental shelf of India. And I would like to propose how they can be used, why they should be used using the work which we have done at National Institute of Oceanography, Goa. So in no way we are negating the work which is done by many people in this field, but which I have already included in some of my publications and review papers. So with this, I will start talking, and I'm very glad that the TMC and Foraminifera Foundation given me a chance to talk to you. And I can see a lot of stalwarts of Foraminifera in the list of the scientists who are attending the lecture today, and they are the one from whose paper I learned a lot of things before. So most welcome. Friends, the relationship of human being with water is inseparable. As the life started from ocean or from the water, and most of the human civilization grew near to the water. It could be the river banks and as well as the coastal areas all over the world. I just downloaded some data from the internet. It's difficult to get a very correct picture, but coast became a very favorite place for the development and concentration of human population. And it is said that about 200 million people worldwide are living along the coastline, which they defined as 5 meters above the sea level. And they say by 25th century, it should be around 400 to 500 million. Now, this development has brought the pressure on the coastal areas. And since this became a preferred area for settlements of the human being, development of cities, as well as a lot of industries. And with the development, the pollution started, and which became a source of conflict. India is no exception. See, you can see how many villages are along the coast, and these villages are the fishing villages. So basically, the living of a lot of coastal people depends upon the catching the fish from the coastal areas. But development of industries and the cities along the coast has created a conflict of interest between those who are interested in development and those whose livelihood is dependent upon the sea, particularly catching the fishes. Therefore, because a lot of industries and the cities, they are polluting the coastal areas. So there is a need to monitor the health of coastal marine water. The question is how to monitor, why to monitor, and whether it is possible to prevent the pollution in the coastal areas. Very first, what is the definition of pollution? And so the word marine pollution is used where normal marine environment is changed due to the man's action. So it is basically the man. There are natural environmental parameters also keep changing, but that is slow. But when man start interfering into the environment, it creates pollution. The history of Just a minute. Yeah. The origin of marine pollution studies in India, it started in 1974, when in Goa, Zuari Agro Industries was established. And soon after the establishment of the industry, the rampon castes, the traditional fishermen, started protesting because they believe that this factory is discharging and its effluent is rich in arsenic, which is killing their fishes. And this agitation brought the point into consideration what need to be done. Factory cannot be closed, but at the same time, we cannot allow the people to pollute that area and take away their livelihood. In 2013 also, there's a newspaper, the minister was remembering that this was India's first ever environmental agitation held in Goa. So when such situation comes, the first question comes, the factory people always say, or the domestic municipal corporations say they are not polluting. But the local fishermen and those who are life depend on the coastal region and they say they are polluting. So someone has to decide that. So what are the tools to decide whether any factory or any domestic waste is polluting or not? There are two types of tools are available and they are easy one. Normally people use the biological tool, the fishes. They take the effluent, put the fishes there and see how they are reacting. But friends, when the pollution is able to kill the fishes, it means it has reached to the irreparable level. What is the fun of telling now it is pollution when everything is dead? We need something which can start, which can warn you in the beginning itself. The other thing is the chemical tool. You can take the samples from the effluent near point and analyze in the lab, say mercury, and this is very easy. But the problem is, if you use the chemical tool, sometimes you end up giving a wrong report. How? If chemists, if you're going for the sampling and factory stop its pollution for say 1 month, effluent or stored within the factory, you will go and sample. By the time it gets diluted and you will give a report that there is no pollution, which will not be a factual report. Okay, or the factory will arrange or manipulate in such a way that you go and take the sample in monsoon time when the coastal water is diluted because of freshwater influx, and you will say that pollution is not of alarming level. To counter it, you can collect the sample fortnightly or monthly, but then project became very expensive, as well as it will take 1 year to give a report, which is not very practical. So in this scenario, when chemical tools and biological tools are having limitation, we can supplement these studies with another tool called foraminifera. So why foraminifera? See, there are many groups, and depending upon the location of, we can take, I will explain the things with the help of foraminifera. This gathering, I not actually bring the slides about the type of foraminifera, but they have been used extensively now in pollution studies because of their fossilization potential. Once a factory is discharging this effluent and it affects the foraminifera, and foraminifera test incorporate its effect. After that, even if factory is closed, we can give a report factory was polluted. So it is the environmental sensitivity, particularly in coastal area of benthic, that can give a very good tool. I just, for the students, if they are there, those who are not from foraminifera, it's planktonic and benthic foraminifera. And I put few slides here. The beautiful shape of foraminifera has been used to develop the park, the ornaments, the lollipop, and so these are the commercial applications which are being exhibited in China. So today we are going to use exhibit how benthic foraminifera can be used. This has been on the Foraminifera Cushman Foundation platform also. These figures are available. Because of this sensitivity, we have used the foraminifera into a variety of applications. They are paleoclimate, sediment transport, petroleum exploration, particularly in our area, monsoon variation, which largely control the agriculture, fisheries, some of the commercial application, the designing, how to get the record of past tsunamis, sea level changes, and sea level changes and monsoon together give an idea how foraminifera can use in the marine archaeology. And we'll talk today about the pollution monitoring. But foraminifera can be used in all these applications. So let us see how they're useful in pollution monitoring. As I told you, the pollution studies started in India in 1974 when first environmental agitation was there. And two people took the lead. One was Dr. M.G.A.P. Sethi. He was trained in Utah. And then he was leading the group at National Institute of Oceanography, Goa. Another was my PhD guide, Professor Bhalla, who was doing it at Aligarh. And these are the publications brought out from the Zuari agro industries starting from 1976. How to collect the samples from the marine environment. Many of you who will be working on the land foraminifera, continental records. From marine records, we have to use grab sample when we are collecting the samples from the bottom of the sea. And subsurface samples could be collected based on the coring. So it is the sensitivity of foraminifera which we use it, and three types of sampling we can use for pollution monitoring. Surface sediment studies, short course, and sampling with the time gap. In any area which is adjacent to the industries, pre-industry and post-industry samples, if we have, we can get an idea. And this is how we collect the samples from the offshore area. This is the sea and this is the land. We get the sediments either by water through river or by the wind. There are microfossils which are living in the sea, and after their death, they're getting deposited at the bottom of the sea, layer by layer, year by year. And this serves as a pages of the history, environmental history of that area. We can take our ship and take a core, put a date on it, and with the help of ecology of foraminifera, we can tell what was happening in the last few years to few million years in any area. Now, so we know that how to collect sample. Now the next question is where to collect the samples. If this is a factory source of pollution, you can collect a grid pattern samples where the effluent is being discharged. So obviously the sample which is having close affinity, they will be affected more. Then we can collect a short core, and if we can see the pre-factory and the post-factory, we can find out. So sample which are affected here, actually this sample and this sample should have the same type of foraminifera. But because this is closer to the effluent source point, they will show the effect of pollution in these samples as compared to these samples, as we move away from the source. So the surface sediment and the core samples will give you an idea how foraminifera are used. So what type of changes we are expecting? When the modification of individuals, the moment any area started getting polluted, the first thing which get polluted is the few individuals get affected. They show the sign of corrosion or morphological abnormalities. These are the very early signs. Then afterwards, when the intensity increases, we get either some species, opportunistic species, take the pollutant as a food in case, and they grow more in number, or they grow in less in number if the effluent is toxic to them. And when the entire fauna is affected, low or high total foraminiferal number we get as compared to the normal environment, and the size of a specimen, normally we see the dwarf specimen in the polluted areas. So With this concept, we did the initial sampling way back in 84, which started actually in 76, but in 84, we brought out first review paper. It's near Bombay, Goa, Karwar, and Trivandrum. It's along the Indian coast. Here is a agro factory which was discharging the effluent rich in arsenic. This is a paper mill factory at Karwar. Here the factory was titanium mineral verification factory. And this is near Bombay, the Thana Creek, where 20 factories were almost throwing their effluent into the small creek. So these 4 areas we have taken and see what type of results we are getting. The individuals get affected. See, this is Spirolochlin developed 2 mouths. This is Virgulina, which develop abnormal growth. All the specimens, this is a normal specimen, and these are all abnormal specimens. Then the second, when the pollution level grow further up, almost all the specimens you can see, they're showing a very abnormal growth. If you compare it with the normal specimen, either chambers are becoming small or too big, or the coiling go in a different direction. So the moment you start getting abnormal tests, that is the sign that pollution already started working in this area. Some species like ammonia, they reduces when the pollution, particularly organic pollution was there, or domestic pollution was there. And some species, opportunistic species like Stenforthia has increased. So in normal, per gram of sediment, if you are likely to get 10 specimens, you get either more or less of these specimens. And then when the entire fauna is affected, see here, almost every species specimens, you can see they are abnormal in nature. And when acidic pollution is there, you can see the corrosion. All the outer level, wherever the soft sediment was there, it got dissolved. And the worst example, I say best example, but in fact, it's a worst example in Thana Creek area. When we sampled this area, we didn't find a single living specimen. Whatever was there, we get a lot of dead specimens, but no living fauna was there. It means that things have gone beyond the repair level. Now, when we published these papers, a series of papers we published, the main objection was there that since you are going to a known polluted area, and whatever abnormality you are noticing, you are attributing it to the pollution, which may not be the case. There may be some other factors which is affecting, and you guys are trying to attribute it to the pollution, so it cannot be the model studies. So we decided, let us see if it is possible to do it into the culture experiment, where of course, no culture experiment can mimic the natural environment in totality, but that is the best way to provide a supporting evidence about what we have seen in the field. So we developed the culture experiments. I learned the culturing. I can see the Howard Sparrow was there. We learned the first thing, culture experiment at Woodsole Oceanographic Institution. And when I came back, I tried to establish a lab at National Institute of Oceanography, which is the only lab which is culturing in Southeast Asia. And we are very hopeful we'll bring out more and more papers through culture experiment, which will be helpful in developing the new proxies, checking some of the old proxies for climatic reconstruction, as well as application of foraminifera in pollution studies. So this is one photograph I put it of our the foraminifera culture laboratory. It's now well established. Almost my entire last 10-15 years have gone up to establish this lab. Now one of my old students is Dr. Rajiv Nigam, is the in charge of this lab. It's all incubators, flow, different type of microscope, water bath, and everything is there. So what we did actually to support our field-based finding that pollution largely bring the abnormality, morphological abnormality in foraminifera test. So we took the specimen field, we grew them into the lab, and you can see these are the large, small babies coming out of the parent specimen. Now we have taken these babies and put into the different experiment. And now, now what we did the mercury experiment. We introduced the mercury in a phased manner. Slowly we have increased, and you can see that it is giving a very good result. This is a normal specimen where no mercury was added into the media. And this is the specimen which grew when mercury was added. So most of the specimen where mercury was introduced into the media, they develop the abnormalities. At the same time, their growth is retarded. This is what we told when we did the field-based experiment. We get more abnormal test and we get the dwarf fauna. Then we have sudden introduction of the different concentration of mercury. And you can see that many specimens had died and almost 100% population, they got affected. This is a normal specimen and this is the abnormal specimens. So the culture experiment has proved or extended the support with our previous findings that pollution brings the abnormality in foraminifera, and that can be taken as a very good tool, because being at the very beginning of the food chain, if foraminifera can be taken, studied, and given a report, we need not wait when the pollution go a very high level and fishes are affected. Even if factory is trying to deceive the investigator, the pre-factory survey will give you a normal specimen, and post-factory specimens which are abnormal can give a report that yes, factory is polluting or not. So foraminifera are proving that they are very good tool to first give a report whether anywhere pollution is there or not. We publish a series of paper into Marine Pollution Bulletin. And one of our students worked into Michael Kusera's lab at VN Linshi. And when she came back, she conducted this mercury experiment and she got the Young Scientist Award by Indian Science Congress in the field of environmental sciences. So people, why I put this slide, we are saying that people started now believing that just foraminifera can be a very good tool for pollution detection. Now the next question, another method is there. If you have the report from the different times from the same locations. This happened in one of the river estuaries in Goa region. This estuary draws its water from the catchment area where a lot of mining activities, iron ore mining is going on. So during the monsoon, when very heavy rain, this region received a lot of material come to the sea and create a pollution. So what happened, we collected the samples in 1974, then 2001 and onwards, and we compared the three different times samples. You can see the suspended load has increased over the years. It is 70, 80, and 1990. And you can see the total foraminiferal number is coming down. So this is a very clear, we publish a series of papers on this estuarine complex. So if you are having, this can be applied to when factory is being established, best thing to go and take the surface sediment samples in that area. And after factory is working for a few years, we can go and again collect the samples and see the changes and decide whether it is harmful or not. You can see here the data. 200 specimen, then 138 specimen, and the 41 specimens. So this shows that foraminifera can be also used to have adverse effect of mining in the nearby areas. Now, once the pollution is detected, what next? See, we cannot close down the factories because they provide their development, the driver of development, as well as the employment. So we cannot close down the factory, but at the same time, We cannot allow them to pollute where fishes are affected and which affects the livelihood of the many coastal people. So what is a remedy? Remedy is we do the physical oceanography survey where the current pattern is such if you take the effluent through a pipeline and put a diffuser, so all effluent goes towards after treatment, it comes here and it goes into the deep sea where it gets diluted. So its effect is minimum. Now, the idea is this pipeline should be buried into the sediment. Because if it is lying on the surface, it will be damaged by the trawlers working in this area. Now, it should get buried into the sediment if sediment is soft. So we do the survey and we give a route where the pipeline should go. So that, and if soft sediments are not there, then trenching is required, which is a bit expensive thing. So what happened? Now, there is a lot of things required here. So once we laid down the pipeline and what happened? See, these are the seismic records, shallow seismic record. See, these are the two pipelines which were buried into the sediment, you can see. But when we did survey after some time, these pipelines were exposed. It means when we are laying the pipeline, sediment transport history of that area also should be considered. How to get an idea about sediment transport in any area? We can use the sediment tracer, we can use the mineral tracers. But for long-term sediment transport detection, we can also use the foraminifera. How it can be used? We can use simply, see, benthic foraminifera, with the help of their pseudopodia, they serve dual purpose. They collect the food and they anchor themselves and thus resist the movement under the influence of currents. After their death, the soft part get decomposed and the empty test are subjected to the sediment transport. Now, if we compare living and dead, we get an idea that whether sediment is moving or not. So if you take any particular species or group of species and see their percentage into the living, okay? And then see the dead, if 10% is here and in the dead specimen, 10% is here, probably sediment is moving in this direction or sediment is moving in this direction. This concept we tested into a near Bombay area and the results show that the species which is covering the 10% area here after their death, they're concentrated here. It means something after their death is pushing towards this direction. The reverse way, which species which were abundant, 80%, they have been pushed towards this direction. Is it true? Whether it is noticed? Yes. See, the lot of fresh water comes through the northern Sutherland channel, which is pushing the sediment towards this direction. And the waves pushing in this direction and resultant is what we notice is in this direction. And this we have published in continental shelf research that foraminifera as an indicator of sediment movement. So not only the foraminifera are helpful in detecting the pollution, but they are also helpful in mitigating the pollution. So when we started that time, very few workers were working. And I remember when I started in 76 as a student, the paper of George Sigley, which was published in the Caribbean Journal of Sciences on foraminifera pollution indicator. From Indian region, we published the review papers. Still we are publishing. And I'm very happy that now in every field, the lot of paper, this is the paper in last 2 years, which is published using foraminifera for pollution monitoring. So friends, we can say that foraminifera can be effectively used and a lot of work is going on almost everywhere. The time is not very far when we can prove and the fact all pollution control board or those who are looking after the health of the ocean in the coastal areas will start using foraminifera for the pollution related studies. And this will be another good application for the commercial purpose, which will also provide a good funding for foraminiferal research. So in the nutshell, we can say that foraminifera are helpful in detecting the marine pollution and designing the preventive measures. We can prevent foraminifera from incorporating evidence of pollution by stopping the pollutants. Let us strive to make our sea pollution free. I use, this is of my lab, different photographs. This is a culture lab. And these are the students whose work I used for today's presentation, basically the work done by them. And as their representative, as a senior person, I'm presenting this work to you. Thank you very much. I was given about 35 minutes, so I hope I have completed within time.
Oh, that's perfect. Thank you very much. Thank you very much. Very nice presentation. Thank you. Actually, there is already, a question by Spiro. So please.
Okay.
Howard, do you have a question? I saw your hand on.
Well, I was actually clapping, but
okay, sorry.
No, that's okay, Rajiv. Really nice talk. I have a quick question for you.
Sure, sir.
Given all of the culture data and the material you've collected over the years to look at morphological change with respect to heavy metal pollution, have you ever had any students go back and analyze some of the calcite just to do a geochemical relationship with morphological change and the actual in situ, say, mercury concentrations or arsenic concentrations that were present that caused the abnormal morphological shapes?
Thank you, sir, for your encouraging remark. This is the future line of research which you suggested. This was bothering us for a long time. We can show that morphological abnormality is there, but pathway is not very clear. This mercury, how it is affecting. We believe that it is affecting the soft part, which finally secrete the calcium carbonate. But big question is whether this pollutant are going and staying into the test of the foraminifera. The facility was not available to me so far. Now, just in last 2 years, a laser ablation technique has been established at our institute. Although I am retired, but my students are still working there. So I will pass on this message, and it will be worthwhile to see the chemistry of the foraminifera test. Other than calcium carbonate, what else going and settling there? Whether pollution is going and settling there? If yes, that will be a wonderful thing to see in past how the things have changed. So yes, geochemistry of foraminifera test. A lot of studies of protoplasm, cytoplasm is required. How pollution actually working? In what is the pathway of taking this effect to the morphology? I think that some papers I have seen on that, but being a geologist, I know very little about biology of foraminifera. So those who come to the foraminifera studies through biology route, I came via geology route. So more interested in morphological changes and abundant senses studies, but it will be worthwhile to study the soft part of foraminifera, take the samples which are from culture experiments and see how that protoplasm is different than the protoplasm where there is no pollution. Same species. Let us leave this question open.
Well, I think the geochemistry question can be answered in a couple of years. The biology question, maybe by 2050.
Yes. Already the work started. People are using the MGCA ratio for paleoclimatic reconstruction. So if they can start using, keeping in mind that how it works in the field of foraminifera as pollution indicator, that will be definitely a very good contribution for by the future workers.
Okay, thank you. We have a question from Pamela Alok.
Okay.
Please.
Hello, madam. How are you?
Nice I'm doing very well, thank you. I recently became aware of an energetics kind of interference in associated with metal pollution. And one of the things that they found was very similar to what we found in bleaching of the forams that caused deformities. And basically what the paper by Toller, I think 1998 or something, so showed that there was a huge deficiency in organic matter in the partly bleached forams, and meaning that they weren't getting enough energy, weren't getting enough carbohydrate from the symbionts. And if the pollutants can interfere with the metabolism of the foram and essentially either interfere with their ability to produce the organic matrix for calcification, that could be a factor, and hopefully that will be explored in the by future culture studies and so forth, because I think that's a possibility there, and it's a very interesting possibility.
Yes, madam, I totally agree with you. And if we wish to propose to the industry that along with chemistry, you should start using foraminifera, we have to answer many more questions, because people will accept only if we come up with all that answers what we are raising today. Because now, I don't know in a Western world how funding is going on, but for foraminiferal studies, now funding is becoming very difficult. So I think that through pollution studies, like earlier, lot of funding for foraminifera research came from the oil industries. Now I think the time we should look forward when the funding will come from the coastal industrial units, and which will give a very impetus to the foraminifera studies. And definitely this metabolism will play a role, because if protoplasm is affected badly by the introduction of the heavy metal or organic pollution, secretion of calcium carbonate will be hampered, and which will finally manifest in form of the morphological abnormality. So I think we all agree that we should encourage our next generation to look into the chemistry of foraminifera trust grown into the laboratory under the controlled condition, and we know that we introduce mercury, where the mercury has gone and how it has affected. That should be the line of study now for the future. We are going to do it at our lab. I request all of you to encourage in other labs as well.
Okay, thank you. So we have now a couple of questions in the chat.
Okay.
I'll read you the first one.
Yes.
It's from Katia Barbosa. She asked, could you observe any salinity influence on morphological abnormalities?
See, salinity otherwise also affect the foraminifera very well in form of the census count of different species. And sometime I will give a lecture on foraminifera and paleoclimatic reconstruction, where we have seen that a lot of waters come through river, and when salinity variation is there, so drought condition and flood condition, they change the population in the nearby river mouth microenvironment, which we use it as a reconstruction of past rainfall records. But as far as pollution is there, yes, some factories are throwing the hot water, because a couple of furnaces in coastal areas, industries, they use the seawater for cooling their instruments, and the hot water is discharged. But definitely it will affect the foraminifera, but I have not studied personally. So, but instead of not only abnormality, I think the total population will change. High salinity species, you will get more in the areas, the vicinity of the coast.
Okay, I see. Actually, I'll go now with a question from the audience, and then I'll go back to the, no, let's go with the chat, sorry.
Okay.
So first there is always Spiro that says hello because he needs to leave. So, and he says a great talk again. Then we have a question from David Barbosa Bergamo that says, I have conducted studies with pollution gradients. I find variations of increase in abundance, richness, and diversity in the intermediate environment of the gradient. The question is, what is the significance of intermediate environment effect on assemblages? Is this a common pattern?
See, the basic thing is you change in any parameter in the environment, marine environment, and you will get a definite signature into foraminifera. Now, these changes could be natural, changes could be anthropogenic. Okay, natural changes affect for a long time and they will not be seen if you take a short core in that area. Okay, so factory introduction of any factory in coastal areas is not more than half a century or 1 century. So if you want to differentiate between the natural changes and factory induced changes, say hot water, you can see through the study of a short core into the vicinity. That will give you a very good answer. And when salinity increases through hot water, you will get the species which is indicating the high salinity, which otherwise will not be available in the nearby areas where hot water is not there, but depth and all other factors are same. Compare it with that few kilometers away, and you will see the difference, which you can list it as an indicator of thermal pollution.
Okay, nice, great. So now we have the hand is raised by Atul, please.
Yeah, I'm Atul. I'm Atul Pandey from Patna University, Bihar, India, and I'm thankful to one of your students, Dr. Nigam Syed Salim, who has joined our university as a faculty a couple of months back. So this is one good news for you and for all of us. And I'm thankful for your very elaborate and very interesting presentation where the complex things of related to foraminifera have been explained in a very simplified manner. My question, sir, to you is that you have mentioned that you have collected samples from the western coast of India, starting from Gujarat, then Mumbai, then Goa, and also from Trivandrum. So you have also talked about the abnormalities in the forums which you have mentioned. So is there any similarity or dissimilarity or the prominence of the abnormalities which you have witnessed in the morphology of the forums from the northern part to the southern part of the coastal plains in the country, in India?
What you said, northern and southern part of?
Means starting from the Gujarat coast to the Trivandrum coast. So the variation from the north to south, is there any statistical or anything special about the morphology?
Yes, there are differences. I think you're talking about the natural differences. Today's presentation I gave about where the factory-oriented industrial pollution or domestic pollution. But there is a changes. Gujarat, if you see, we get less fresh water into the sea in Gujarat, along the Gujarat coast. The heavy water started coming to the coastal area, Bharuch down, when Narmada and Tapti are joining. Okay, so the fresh water, the salinity of the coast is very high if you go towards the Gujarat side. You come to Bombay, it is quite lower. But you come to southern side, still there is no big river. So very small rivers are there. And if you want to see the effect of freshwater, which in fact we are using, Salim has worked on east coast of India for his PhD. So you can compare the west coast and east coast at the same depth. See, they are on the same latitude. So temperature is almost same. What make a big difference is the salinity, because most of the big rivers are draining their fresh water into the east coast of India. So there is a vast difference if you come north to south as well as east to west. And this is a really, some other time when we talk about paleoclimatic reconstruction, probably I will give you more elaborate examples on that. And I'm very happy that you people have got Salim. He's an excellent student of mine. And I'm very proud of my students, I will tell you. 5 of them won the Young Scientist Award. Rajiv Saraswat bagged 5 Young Scientist Award alone. Then Rupal got it for this archaeology. Rajni and Lindsay got it for the pollution studies and studies of Myanmar. We got the cyclicity through foraminifera in climate change. Okay, and based on that, we predicted Nilay Khare's PhD work. We predicted that we will have a better rainfall in next 20 years, and last 20 years prove that what we talked based on simple small foraminifera, now meteorological data support us. That was a big encouragement. So I'm actually a big fan of foraminifera. You give me any problem related to marine environment, probably will find a solution. This, since you are from India, the mud bank problem of Kerala, when fishing is stopped all along the Indian coast because of rough weather during monsoon, Alappi area, about 8 square kilometer area is full of fishes and behave like a pond. This question was debated for last more than 100 years. Why this phenomena? Why this area is not disturbed and why we get so much fishes which is being supplied to the rest of the country during monsoon period. We found based on foraminifera that is a subterranean flow is there from the nearby lake, which brings the nutrient from the bottom and which brings the lot of fishes to that area. So forums, I'm happy you got forum man, encourage him. Forums can give beautiful answer to many of the modern problems, as well as problem of the past, as we talk about application in marine archaeology as well. How that the world's biggest, oldest dockyard we found in Gujarat, which is based on the foraminifera. We solved 50-year-old archaeological controversy with the help of foraminifera. And it is settled now. I'm really glad that my tool is so good.
Oh, thank you. So going back to the previous question, we have a comment from Malcolm Hart that says, we have used the laser ablation, ICP-MS, to study the form of the foraminifera, but while it gives the chemical profile of the shell, it does not identify which element or elements cause the problem.
See, this, if you take the specimen from the field, this problem will come. Because you may think it is because of pollutions, others will say may not be. So best thing is collaborate with the people who are culturing foraminifera. Okay, I gave some samples to Paulowski for doing the DNA studies. We published one paper also on DNA studies of foraminifera. So you take the culture specimen and then do the laser ablation or whatever chemical instruments you want to use in the lab. That will give a better answer. So you know that under what circumstances this specimen grew. Okay, and if you can get back like temperature and salinity, you knew that your specimen is grown at 30-degree temperature and say 35-degree salinity. Now you subject them to isotope analysis and see you get back calculation, same temperature and same salinity. If just tool is perfect, then you can use isotope for the further work. If not, you have to think where things going wrong.
Another question in the chat, then I'll go. Lander Mermamora I hope I pronounced it correctly. Have you seen positive effects on organic pollution on the sites of normally small hypoxic tolerant taxa, maybe like abnormal growth?
Yes. See, particularly when domestic pollution is organic pollution is there, some opportunistic species take over. And sometimes the situation comes, only one species grow on the cost of other species. For example, you are getting earlier 100 specimens and there are 10 species, equally distributed 10% to all 10 species. But you start introducing a pollution in that domestic pollution, only 2 species will be sharing 50-50%. All other species disappeared. So different species behave in a different way as far as the abundance goes. And this we have seen in natural environment also. You start increasing the temperature in any area, the composition will change. So there is only one difference between the natural and anthropogenic changes. Natural changes take a lot of time, whereas anthropogenic changes can bring the difference within a, you can say 1 or 2 years itself.
Okay, now we have a question from Schoenfeld.
Hello, Professor Nigam.
Hello, good morning.
Hello.
I have a question concerning Mumbai Harbor. Your
Okay.
investigations have taken place more than 30 years ago.
Yes.
And my question is, have the foraminifera returned to that creek?
Oh, great. I'll tell you this, great, thank you. This we have done it long back, and when we found that there was no living foraminifera in that area. But Sangeeta Pashkude from Wadia College of Bombay, Wadia College of Pune, she worked on Thana Creek sample and she got some living foraminifera. Because in last 2-3 decades, lot of precautionary measures have been taken. Government of India brought resolution, everyone has to treat their pollution. Then carrying capacity studies also we are doing it, how many factories can come. Earlier, I will tell you a very interesting question you raised. One factory, say 10 tons of effluent it was discharging, and we say carrying capacity, okay, seeing the size of the basin, it is okay. Now the second factory says, we say 10, okay, fine. Collectively, we are actually discharging 100 tons, but each individual factory got permission because they are telling only 10. So finally, we started conducting the carrying capacity of the basin. So that brought out a good report, and then treating the effluent within the factory also is bringing a good result. But you know that when it comes to the money, treatment is a very expensive job. So if there is no continuous policing is there is a tendency to pass on even less treated effluent into the sea. And therefore, that care has been taken by the Pollution Control Board. Now, each state is having a pollution control board, which they are trying their level best. So yes, there is example, foraminifera came back into Thana Creek area.
This
Thank
is
you,
very
sir.
nice. I appreciate this very much. Thank you, Professor Nigam.
Thanks a lot, sir. You brought a very good question, so I can give this new example, which actually was not in my lecture. Thank you, sir.
Okay, so there are several messages in the chat that say that's an excellent presentation, wonderful talk. They thank you, Professor Nigam, for your presentation. So there are people, there are, for instance, Pam Alloc, Katia Barbosa, Luciana Ferraro, Professor, this is difficult to pronounce it for me, Benkata Chalapati, Malacordi, and so on. But I don't see any other questions. So if there are no questions, I want to thank you very much, Professor Nigam. Rajiv,
Thank you. This is our Indian way of saying thank you to you.
For accepting to give this talk. we all enjoyed very much, and we have seen from the many questions that that come from the audience that it was very interesting. This is already very clear. So thank you very much again. So if there are no other talks, other questions, I don't see any other questions. So thank you very much again, and we'll have a next seminar in February. Don't miss it, and hope to see you again here in few weeks. Thank you again, and have a nice.
I also
Thank
would
you.
like to thanks to you giving me an opportunity to talk to a number of people, and very good questions which give us thinking what should be the next to do it. And your committee is doing an excellent job of promoting foraminifera worldwide. And if sometime I get opportunity, I would like to talk on marine archaeology next time. Thank you very much.
Of course, I mean, we plan to have these seminars for a long time in the next years, forever, if possible, forever. So there will surely you can come back again and give your seminars on this topic with no problems. Absolutely.
And also
you're welcome to give it.
And I also extend the welcome all of you to come and see our lab, which is a wonderful culture lab in the South City. This is the only lab in South Asia which is culturing foraminifera. Please do visit us. Thank you very much.
Oh, personally, I would love to come. That can be really great.
We'll
So
find out some reason to come.
I'll try to find a reason to come. So thank you again, and have a nice morning, afternoon, wherever you are in the world, and goodbye.
Okay, bye.
Maria, Maria, I just wish to just request you keep on informing us about the programs